Hello, and welcome to my channel. This video is on the neurochemistry of ego death, how pharmacology dissolves the integrated self. I'm going to begin by going over the science of ego death and the science of consciousness, just to give you some scientific background. However, if you're not interested in that and you want to just get straight to the story of this video, please skip to slide 4. So we're going to go over these three scientific articles I found on ego death, and I'll start with just a summary of each one. This one is called A Novel Ego Dissolution Scale, a Construct Validation Study. So the main question in this one is, how can ego dissolution be measured reliably, and is it distinct from dissociation? The researchers developed a new ego dissolution scale to measure more trait-like or general tendencies toward ego dissolution rather than only psychedelic-induced experiences. They tested the scale in a large participant sample and compared it with measures of mysticism, disassociation, personality, and unusual experiences. The key findings are ego dissolution appears to have two dimensions: ego loss, which is reduction or weakening of the self boundaries; unity, which is feelings of connectedness or oneness. The scale showed strong reliability and validity. Ego dissolution and dissociation overlap but are not the same phenomena. Disassociation tends to involve fragmentation or detachment, whereas ego dissolution may also involve positive unity and self-transcendence. The paper provides a more rigorous psychological tool for studying ego dissolution across psychedelics, meditation, mystical states, and psychopathology. So ego death has to do with a lot more different states of consciousness than psychedelic states. So the next paper is Losing the Self in Near-Death Experiences, the experience of ego dissolution. The main question here is, do near-death experiences involve ego dissolution similar to psychedelic or mystical experiences? The researchers examined reports from people who had undergone NDEs and assessed ego dissolution using established questionnaires. The key findings were many NDEs involved strong loss of self-boundaries and diminished sense of individual identity. Ego dissolution in NDEs correlated with feelings of unity, transcendence, altered perception of time and space, and mystical type experiences. The experience often resembled reports from psychedelic states, especially regarding self-loss and interconnectedness. The paper suggests that ego dissolution is not unique to psychedelics. It may be a broader feature of altered states of consciousness, including NDEs, where the normal sense of self temporarily weakens or disappears. So the next article is my favorite title, Me, Myself, By Regional Alterations in Glutamate and Experience of Ego Dissolution with Psilocybin. The main question here is, what neurochemical changes in the brain are associated with psilocybin-induced ego dissolution? Researchers use psilocybin versus placebo, brain imaging, and magnetic resonance spectroscopy. And they use these three techniques to measure glutamate levels in specific brain regions during psychedelic experiences. The key findings were psilocybin caused region-specific glutamate changes. In the medial prefrontal cortex, they found higher glutamate linked to more negatively experienced ego dissolution. So they found anxious or destabilizing loss of the self. In the hippocampus, they found lower glutamate linked to more positively experienced ego dissolution. So they found unity, peaceful self-loss, and an oceanic boundlessness. The study also found altered connectivity in self-related networks such as the default mode network otherwise known as the DMN. Ego dissolution during psilocybin is associated with measurable neurochemical and network level brain changes, particularly involving glutamate and self-processing circuits. The emotional tone of ego dissolution, pleasant versus distressing, may depend on where those neurochemical changes occur. So the overall synthesis of these three papers is together, ego dissolution is measurable. It occurs across multiple altered states, including NDEs or near-death experiences, not just psychedelic states. And it has identifiable neurobiological correlates, especially under psychedelics, like glutamate being increased or decreased. So moving on, we're going to talk about the mechanism of consciousness. A common theme is that the self behaves less like a fixed entity and more like a dynamic psychological process that can become weakened, reorganized, or temporarily dissolved under certain conditions. Together, these three papers suggest a possible framework in which consciousness may emerge from highly organized and dynamic processes occurring inside neurons, particularly involving microtubules and phase-separated biomolecular networks. The first paper shows that helical biological structures like microtubules can theoretically support collective quantum optical behaviors such as superradiance and subradiance, implying that the cellular geometry may organize coherent energy transfer and signaling. The second paper demonstrates that microtubule associated proteins form fluid fractal-like networks through liquid-liquid phase separation, also called LLPS, highlighting how cells organize information and biochemical reactions through dynamic condensates rather than rigid structures alone. The third paper provides experimental evidence that stabilizing microtubules delays anesthesia-induced unconsciousness in rats, suggesting that microtubule integrity may influence conscious brain states. Taken together, these findings do not prove a complete mechanism of consciousness, but they support the idea that consciousness could arise from coordinated processes spanning molecular condensate, cytoskeletal organization, and large-scale neural activity, where dynamic coherence and multi-scale biological organization contribute to the emergence and maintenance of conscious experience. I just want to walk you through all of these pictures just so you definitely know what they are. If you want to look at these papers, you can definitely read more details. But right here, they're just showing the tryptophan is in blue on the microtubules, the actin, and the amyloid fibrils. The microtubules are found inside the cell, actin is found inside the cell, and amyloid fibrils are found mostly outside but can be found inside the cell. And these tryptophan moieties attach to these proteins or aromatic so that they can mediate the superradiance. And the superradiance is a quantum mechanism that can utilize the information that is being integrated into the system to build a holographic map of reality to generate the conscious experience. So it needs to be dynamic because our day-to-day experience is a fluid experience and it's always changing. So all of these proteins are intrinsically disordered, which gives them the flexibility to integrate that information as it's constantly coming in. So most proteins are well-folded proteins, as you can see here, but these proteins associated with superradiance organize via disordered, via being intrinsically disordered. So unlike most proteins where they just have one fixed shape, intrinsically disordered proteins have an ensemble of conformations, and that allows them to aggregate together and remain flexible and dynamic so that they can take incoming signals and integrate them and change as new signals come. And that's very important to have that flexibility for these quantum states to occur and to be able to evolve through time. And I also put here, this is a picture of galectin-3 condensates in green and fibronectin fibrils in red, because I did want to show that this happens outside of the cell. And I wanted to show this is a picture that I took whenever I was doing research in lab to show that I do know a few things about these molecular interactions. And we're going to move on to the next slide, which is also a paper I wrote along with my previous boss, Dr. Paul Sylvester, and this was in the first edition book of Quantum Mechanics of Biological System, and it's called Chaos to Coherence because I mainly talked about these intrinsically disordered biomolecules that functioned in between the critical point between chaos and coherence, where you had enough structure for everything to be held together, but also flexibility so those quantum effects could be exploited. So I'll just go through this is the abstract. If you want to read the details, please find this on the web and you can read it, read more of the chapter and get the details. However, I will suggest that you read this whole book because the whole book is a great introduction to quantum mechanics of biological systems. So it reads, recent scientific discoveries have led to the emergence of novel research in the field of quantum biology. Current models of quantum gravity have enabled researchers to build a dynamic framework to understand an approximate representation of the energy landscapes for systems operating at various scales. And the quantum gravity frameworks I'm referring to here is loop quantum gravity, and also string theory. And the type of string theory I'm most interested in is dodecahedron string theory. So you can look that up on the web too if you're interested in reading more about that. And it continues to read, specifically, these models estimate probabilistic energy landscapes by utilizing polyhedral lattices that have similar structure at different scales. Theoretically, information can traverse from one scale to the next, which enables computational cycles to emerge, which is percolation theory. And percolation has been shown to regulate these condensates that are mediated by the intrinsically disordered molecules. And the paper on the previous slide that connected these condensates to fractal organization also found that it was that percolation was the mechanism for this fractal organization. So it's all connected if you would like to read more of the details here. So biological systems also display polyhedral geometry at different scales. Therefore, a quantum framework could offer insights into the balance between chaos and order that is utilized during evolution as a complex adaptive system. This evolutionary system is embedded in a continuous quantum network composed of entangled sections of space that generate energy landscapes such as magnetic and electromagnetic fields. The present chapter will describe how life functions as an agential material guided by holographic superradiance, which enables adaptation to the environment and maximizes the utilization of randomly acquired traits throughout time, aka evolution. This dynamic network can be simply visualized as a lattice composed of emergent patterns and fractal chaos within a dynamic dodecahedron fractal framework. I talked about fractal frameworks a lot in my previous videos, so if you want to learn more about what a dynamic dodecahedron fractal framework is, please look at my previous videos. So moving on to the story now. We're done with all the scientific literature. However, I do think it's very interesting. If you like reading, you can read more of the details in the articles I have presented here. So, what if your sense of self, your memories, identity, and inner voice was not a permanent thing at all? What if the you that feels so solid is actually a carefully organized biological process, one that chemistry can temporarily dissolve? People call this experience ego death. It has been reported during psychedelic states, deep meditation, near-death experiences, and certain neurological conditions. Many describe it as becoming one with everything, while others experience fear, disorientation, or complete loss of self-boundaries. But what is actually happening inside the brain? We explore the neurochemistry of ego death and how pharmacology may temporarily dismantle the integrated self through neural networks, liquid-like biology, and fractal organization. The first thing to understand is this: your ego is not a single structure in the brain. There is no tiny self-center hiding behind your eyes. Instead, the self emerges from coordinated activity across many interacting systems. Neuroscientists often point to a network called the default mode network, or DMN. This network includes regions involved in autobiographical memory, self-reflection, and constructing the narrative of me. The DMN helps answer questions like, who am I? What happened to me? What do others think of me? What story connects my past to my future? Under ordinary conditions, this network helps integrate experience into a coherent identity. But coherence is not rigid. The self depends on constant communication and dynamic organization. And this is where things become fascinating. The brain is often imagined as electrical wiring, but biology is not just wires, it is also fluid. Inside neurons, many proteins and RNA molecules organize through a process called liquid-liquid phase separation, or LLPS. Rather than being enclosed by membranes, these molecules form liquid-like condensates, tiny droplets that concentrate information and biochemical activity. These condensates help organize RNA metabolism, protein translation, synaptic signaling, cellular stress responses, and much more. Instead of rigid compartments, life often uses dynamic droplets that assemble, dissolve, and reorganize. This matters because cognition itself depends on organized molecular states. The brain is not merely firing neurons, it is an evolving hierarchy of organized matter, from molecules to cells to circuits to consciousness. So how can pharmacology dissolve the sense of self? Classic psychedelics like psilocybin and LSD primarily activate the 5-HT2A serotonin receptor. This receptor is highly expressed in cortical regions involved in perception and self-processing. Activation of 5-HT2A receptors appears to increase neural flexibility and disrupt ordinary communication patterns. Brain imaging studies often show decreased integrity and synchrony within the default mode network during psychedelic states. The brain becomes less constrained, more connected, more entropic. Networks that normally remain separated begin communicating in unusual ways. The result, the narrative self loses its ordinary boundaries. Past and present blur. Internal and external distinctions weaken. The feeling of being a separate observer may dissolve entirely. This does not necessarily mean consciousness disappears. Instead, the organization of self-related processes changes. The integrated ego becomes temporarily destabilized. This is where fractal organization offers an interesting framework. Fractals are patterns that repeat across scales. Nature uses fractal architecture everywhere: trees, blood vessels, lungs, neural networks, even galaxies. The brain also displays fractal-like dynamics, nested patterns of activity interacting across many levels. Your identity may emerge from this hierarchical organization, not from one location, but from coordinated patterns repeating across scales. Molecular organization influences cellular behavior, cells influence circuits, circuits influence cognition, and cognition reinforces the self. From this perspective, the ego becomes a multi-scale emergent pattern, a living fractal of organized information, and pharmacology may temporarily loosen the coupling between these scales. The pattern does not vanish forever, but its integration becomes altered, like a whirlpool losing its shape while the water remains. Science has firmly established that liquid-liquid phase separation organizes boundary conditions in biology without the need for membranes. LLPS helps regulate the molecular architecture supporting neural communication and adaptability both inside and outside. If cognition depends on dynamically organized molecular environments, then condensate behavior influences how stable or flexible brain states become. This does not mean droplets alone create consciousness. There is also the well-known mechanisms of genetics and bioelectricity. However, it does suggest that consciousness may emerge from layers of organization that are more fluid and dynamic than we once imagined. Ego death challenges one of our deepest assumptions, that the self is fixed. Neurochemistry suggests otherwise. The self may be an organized process stabilized by neural networks, supported by molecular organization, and shaped through fractal interactions across biology. Pharmacology does not necessarily destroy consciousness. It may temporarily reorganize the architecture that gives rise to the feeling of I. And perhaps that is the deeper lesson. The self may not be a thing we possess, but a pattern we continuously become and evolve. If consciousness emerges from dynamic organization, what happens when the pattern changes? If you like this video, please like and subscribe and explore the science and philosophy with us. And always remember that the more you learn about yourself, the more you can understand others. Have a good one.